somewhere beneath the wet, leaf-littered earth of suburban Sydney, tucked inside a silk-lined tunnel no wider than a human thumb, something ancient is waiting. It has been waiting for a very long time. Not minutes, not hours, millions upon millions of years. Its ancestors stalked the undergrowth when dinosaurs still ruled this continent. Its fangs can punch through a human fingernail. Its venom can stop a grown man's lungs in under 3 hours. And the truly unsettling part, it lives in one of the most densely populated cities on Earth, wedged between garden rockeries and swimming pools and the undersides of suburban laundry piles. It is glossy, black, aggressive, and when provoked, it does not run. It rears up on its hind legs, bears a pair of fangs that drip with venom, and it strikes. Again, and again, and again. This is the Sydney funnel web spider, Atrax robustus, the most dangerous spider on the planet. Now, before you click away, here is the fact that should root you to this screen. This spider's venom was never designed to kill you. Not even close. Primates did not exist when the funnel web's chemical arsenal was being forged by evolution. Somewhere between 150 and 200 million years ago, the lethality of this venom to humans, to us, is a pure accident of molecular biology. An evolutionary coincidence, so staggeringly unlikely that researchers have spent decades trying to figure out how a 42 amino acid peptide, crafted to deter lizards and bandicoots, ended up being one of the most efficient human killers in the entire animal kingdom. Dogs can shrug off a funnel web bite. Cats barely flinch. But a single male funnel web, no bigger than a child's fist, can put a healthy adult human into respiratory failure and cardiac arrest, sometimes in as little as 15 minutes. And that male spider, he is not hunting you. He is looking for a girlfriend. That is the darkest twist in the whole story. The most dangerous encounters between humans and funnel web spiders happen during the warmer months, when sexually mature males abandon their burrows and wander, often for days, across open ground, through gardens, into garages, across pool decks, and occasionally right into a pair of shoes left outside overnight. They are desperate, exposed, and loaded with the most potent version of their venom that their bodies will ever produce. If you startle one, or step on one, or accidentally trap one against your skin, you will understand immediately why this spider has earned its reputation. The bite is excruciating. The fangs are huge, angled downward like twin daggers, and the spider does not let go easily. It grips, it bites repeatedly, and it delivers a full payload of neurotoxin with almost every strike. Between 1927 and 1980, 13 people died from confirmed Sydney funnel web bites. 7 of them were children. One was a toddler who was bitten on the finger while sitting on the laundry steps of his family home in the Sydney suburb of Thornley. He died later that evening. That was 1927, and it was the first recorded human fatality attributed to this species. It would not be the last. For over half a century, a bite from a male Sydney funnel web spider was, in many cases, a death sentence. There was no antivenom. There was no reliable treatment, and the spiders lived, quite literally, in people's backyards. Then, in 1981, everything changed. But we will get to that. For now, settle in. Dim the lights. Let the hum of the night settle around you. Because this is the story of a spider that has been here far far longer than we have. A spider whose body is a masterwork of evolutionary engineering, whose venom holds secrets that could one day save millions of human lives, and whose relationship with the city of Sydney is one of the strangest, most dangerous, and most fascinating stories in all of nature. This is the Sydney funnel web. Let us go back, way, way back, to understand what the Sydney funnel web spider is and why it is built the way it is. You need to understand where it comes from. And that story does not begin in Sydney. It does not even begin in Australia. It begins in the ancient oceans of the Paleozoic era, more than 400 million years ago, when the ancestors of all arachnids were still aquatic chelicerates, animals related to horseshoe crabs and sea scorpions, crawling along the floors of shallow seas. The transition from water to land was one of the great dramas of early animal evolution, and the ancestors of spiders were among the pioneers. By the Silurian period, roughly 440 million years ago, the first arachnid-like creatures had hauled themselves onto shore. They were thick-bodied, 8-legged, and equipped with book lungs, specialized respiratory organs that allowed them to breathe air. These were not yet spiders. They were trigonotarbids, an extinct order of spider-like arachnids that shared a superficial resemblance with modern spiders, but lacked the two features that would come to define them: spinnerets for producing silk and venom glands for subduing prey. Trigonotarbids were ground-dwelling predators that thrived for over 100 million years in the vast club moss and fern forests of the Carboniferous. They were successful, but they were an evolutionary dead end. They left no living descendants. The first true spiders appeared around 380 million years ago during the Devonian period. Fossils like Atrax fimbriunguis, discovered in ancient rock deposits in New York State, represent some of the earliest known animals with the defining characteristics of spiders: a narrow waist separating the two main body segments and structures associated with silk production. These proto-spiders were still primitive. They did not build webs the way modern spiders do. Their silk was likely used for simpler purposes: lining burrows for protection, wrapping eggs to prevent desiccation, perhaps laying down simple trip lines on the ground to detect the vibrations of passing prey. But the foundation was there. Silk and venom, the two great innovations of spider evolution, were taking shape. Over the next 100 million years, as the Carboniferous gave way to the Permian, spiders diversified dramatically. The earliest segmented spiders, members of the group Mesotheli, spread across the forests of the supercontinent Pangaea. These spiders had their spinnerets positioned underneath the middle of their abdomen, not at the rear like modern species, and they were almost certainly ground-dwelling ambush predators, hiding in burrows and waiting for cockroaches, silverfish, and other early arthropods to wander within reach. Then came a crucial split. Sometime during the late Paleozoic or early Mesozoic, more than 250 million years ago, spiders diverged into two major lineages that still exist today. The first was the Megalomorphi, the so-called ancient spiders, a group that includes trapdoor spiders, tarantulas, and, critically, the funnel web spiders. The second was the Aranomorphi, the true spiders, which today make up the vast majority of spider diversity, from orb weavers to jumping spiders to black widows. The megalomorphs retained many of the ancestral characteristics of early spiders. Their fangs, called chelicerae, strike downward in a parallel stabbing motion. Rather than crossing over each other in the pincer-like action used by aranomorphs, this downward strike is enormously powerful. It allows megalomorphs to drive their fangs through tough materials, through beetle shells, through the skin of small vertebrates, and, notoriously, through human fingernails and the soles of soft shoes. Their venom glands are large and sit entirely within the chelicerae, giving them ample storage for their chemical weapons. And their bodies are robustly built, with strong legs and a heavy, muscular cephalothorax. These were spiders built for power, not speed for ambush, not pursuit. The oldest known megalomorph fossil is Rosomigel, discovered in Triassic deposits in France, dating to approximately 240 million years ago. This spider was tentatively placed in the family Hexathelidae, the group that once included the funnel-web spiders. Though recent taxonomic revisions have reclassified the funnel webs into their own family, Atracidae, molecular clock studies suggest that megalomorphs may have originated even earlier, possibly during the Carboniferous, over 300 million years ago. That means the ancestors of today's funnel-web spiders were already diversifying when the first dinosaurs were just beginning to appear. By the Permian period, around 270 million years ago, fossils show spiders with elongated spinnerets, the kind of anatomy associated with funnel web construction. Researchers believe that the funnel and sheet web, a style of web in which a broad sheet of silk funnels into a tubular retreat where the spider hides, was likely the ancestral web type for all megalomorphs. This is the exact architecture still used by the Sydney funnel web today. In other words, the fundamental design of this spider's hunting strategy has remained largely unchanged for 250 million years. It was already working before flowers existed, before birds existed, before the continents had even finished separating into the land masses we recognize today. It is worth pausing here to consider just how successful this body plan has been. The megalomorph spiders have survived 3 of the 5 great mass extinction events in Earth's history. They persisted through the end Permian extinction, the worst ecological catastrophe the planet has ever experienced, which wiped out roughly 90% of all marine species and 70% of terrestrial vertebrates. They survived the end Triassic extinction. They were here when an asteroid slammed into the Yucatan Peninsula 66 million years ago and ended the age of the dinosaurs, while the planet's dominant animals rose and fell. These spiders kept doing what they had always done, sitting in their silk-lined burrows, reading vibrations through their trip lines, and striking with those downward driving fangs. The fundamental design worked. Evolution saw no reason to change it. The molecular evidence tells us something equally remarkable about the funnel web's chemical weaponry. The genes encoding the delta hexatoxins, the family of venom peptides that includes the lethal delta atracotixin, show extraordinary sequence conservation across funnel web species, far more than would be expected for genes under typical evolutionary pressures. This suggests that these toxins have been under strong purifying selection, meaning that any mutations that altered the toxin structure were quickly weeded out because the original version was so effective. The basic molecular blueprint for the Sydney funnel web's killer venom has been essentially unchanged for at least 150 million years, and possibly much longer. To put that in perspective, when these toxins first appeared, the Atlantic Ocean did not yet exist. South America and Africa were still joined. The first flowering plants were just beginning to evolve, and yet the molecule that can kill a human being in 15 minutes was already perfected. When the ancient supercontinent Gondwana began to break apart, Australia drifted northward as an island continent, carrying its cargo of megalomorph spiders with it, isolated from the rest of the world's spider populations. For tens of millions of years, the Australian funnel webs evolved along their own unique trajectory. The family Atracidae, which contains all Australian funnel web spiders, is found nowhere else on Earth. It currently comprises 3 genera: Atrax, Hadronich, and Illawarra, with around 35 described species, all of them confined to eastern Australia. But of all these species, one became the most infamous. One found itself sharing its habitat with the fastest growing city on the continent. Atrax robustus, the Sydney funnel web. Now, let us look at this spider up close. The Sydney funnel web is a medium to large spider with a body length ranging from about 1 to 5 centimeters, which is roughly half an inch to 2 inches. That does not sound especially big, and it is not compared to some of the continent's tarantulas. But what the funnel web lacks in sheer size, it makes up for in sheer menace. Both males and females are glossy and darkly colored, ranging from a deep, almost iridescent blue-black to shades of dark brown or plum. The carapace, the hard plate covering the front section of the body, is smooth, almost hairless, and so polished that it gleams wetly in the light. It is the kind of surface that looks almost liquid, as though the spider has been dipped in crude oil. The abdomen, by contrast, is covered in a fine layer of velvety hairs. In females, the abdomen is plump, rounded, and considerably larger relative to the cephalothorax. In males, the abdomen is slimmer, more elongated, and the legs are noticeably longer. The average leg span for a Sydney funnel web is around 6 to 7 centimeters, though specimens have been recorded with leg spans approaching 10 centimeters. At the rear of the abdomen sit the spinnerets, the silk-producing organs that are a hallmark of this spider. In the Sydney funnel web, the spinnerets are unusually long and finger-like, extending conspicuously from the rear of the body. They are one of the easiest ways to distinguish a funnel web from the many other large, dark spiders that share its habitat, including mouse spiders, trapdoor spiders, and black house spiders, all of which are frequently mistaken for funnel webs by startled homeowners. But the feature that commands the most attention, the feature that has earned this spider its fearsome reputation, is its fangs. The chelicerae of a Sydney funnel web are massive relative to the spider's body size, projecting forward from the head like a pair of curved black hooks. These are not the delicate pincers of a garden spider. These are industrial-grade puncture weapons, capable of penetrating a human fingernail, piercing through leather gardening gloves, and driving deep into flesh. Each fang sits at the tip of a robust chelicerae base that houses the venom glands. And when the spider strikes, those fangs swing downward in a powerful, parallel arc, driving into the target with remarkable force. Unlike the aranomorphs, whose fangs work in a horizontal pinching motion, the megalomorph fang strike is a vertical stab, and the muscles that power it are disproportionately strong for the spider's body size. Males possess an additional distinctive feature: a conical spur or projection located on the underside of the middle segment of the second pair of legs. This tibial spur is a characteristic of the genus Atrax and is used during mating. The male uses it to hold the female's fangs apart during copulation, preventing her from delivering a lethal bite while he transfers his sperm. It is an elegant, if harrowing, solution to the problem of mating with a partner who could kill you at any moment. The eyes of the Sydney funnel web are clustered in a tight group at the front of the carapace. 8 small, dark eyes arranged in 2 rows. They do not see well. Like most megalomorphs, funnel webs rely far more on vibration and touch than on vision. The fine sensory hairs covering their legs and body are exquisitely sensitive to the faintest disturbances in the air and ground, allowing the spider to detect the approach of prey, predators, or potential mates with remarkable precision. A funnel web spider sitting at the entrance of its burrow is essentially a living seismograph, reading the vibrations that travel through its network of silk trip lines the way a musician reads the notes on a page. These sensory hairs, called trichobothria, are so fine that they can detect air currents created by the wing beats of nearby insects. Each hair sits in a tiny socket that allows it to pivot freely, and when even the faintest vibration moves it, the hair triggers a nerve signal that the spider's brain processes with startling speed and precision. In complete darkness, deep underground, a funnel web can pinpoint the location, size, and direction of travel of an approaching animal using nothing but these hairs and the vibrations transmitted through its silk. The spider's respiratory system is another testament to its ancient lineage. Like all megalomorphs, the Sydney funnel web breathes through 2 pairs of book lungs, layered respiratory organs located on the underside of the abdomen. Each book lung consists of a series of thin, stacked plates, resembling the pages of a book, through which air circulates and gas exchange takes place. This system is efficient for a sedentary, burrow-dwelling animal, but it has a critical weakness: it loses moisture rapidly. A funnel web exposed to dry, warm air will dehydrate far more quickly than a similarly sized aranomorph spider. This vulnerability to desiccation is one of the primary factors shaping the funnel web's ecology, behavior, and habitat preferences. It is the reason these spiders are nocturnal, the reason they build burrows in moist soil, the reason they avoid open ground during the day, and the reason they seem so drawn to the watered, irrigated landscapes of suburban Sydney. The digestive system of the funnel web is relatively simple compared to some other arachnids, but no less effective. Like all spiders, funnel webs practice external digestion. After subduing their prey with venom, they regurgitate digestive fluids onto or into the captured animal, liquefying the soft tissues, and then suck the resulting nutrient slurry back through their narrow esophagus. The process can take hours, with the spider crouching over its meal in the darkness of its burrow, patiently extracting every usable calorie. Prey items include beetles, cockroaches, insect larvae, land snails, millipedes, and occasionally small vertebrates like frogs and lizards. The diet is opportunistic. The funnel web will eat whatever blunders into its trip lines, and it rarely passes up a meal. Speaking of that burrow, Let us go underground. The name funnel web is not a poetic invention. It is a precise description of the spider's engineering. The Sydney funnel web constructs a silk-lined tubular retreat, usually in moist, sheltered locations. Under rocks, beneath logs, inside crevices in garden rockeries, within the root systems of trees, or tucked into the gaps between the foundations of houses and the surrounding soil. The burrow itself can extend 20 to 60 centimeters into the ground, depending on the soil conditions and the age of the spider. It is lined throughout with a dense layer of creamy white silk that serves multiple functions. It waterproofs the burrow. It maintains humidity. It provides structural reinforcement. And it acts as a sensory network, transmitting vibrations from the surface down to the spider waiting below. At the entrance of the burrow, the silk fans outward into a distinctive funnel or collar shape, sometimes splitting into a Y-shaped or T-shaped opening. Radiating outward from this entrance are irregular trip lines, strands of dry silk laid across the ground surface in a seemingly haphazard pattern. These trip lines are the spider's alarm system. When an insect, a beetle, a cockroach, a millipede, a small lizard, or even a land snail blunders across one of these strands, the vibration races down the silk and into the burrow. The spider, which has been sitting motionless in the darkness of its tunnel, its front legs resting lightly on the silk walls, detects the disturbance instantly. What follows is an explosion of movement. The funnel web launches itself from its retreat, racing up the tunnel and bursting from the entrance with its fangs raised. The prey is seized, bitten, envenomated, and dragged back into the burrow to be consumed. The entire attack, from detection to capture, can take less than a second. After heavy rain, funnel web activity spikes dramatically. Rain saturates the soil, and burrows that were once snug and dry can flood within hours. When this happens, the spiders are forced to the surface, and suddenly they are everywhere, clinging to walls, crouching under overturned flower pots, sheltering beneath the eaves of houses. This is when bites are most common. And it is also when the Australian Reptile Park's Spider Donation Program sees its biggest influx of captured spiders from the public. The distribution of funnel webs across the Greater Sydney region is surprisingly uneven, and it correlates closely with soil type, vegetation cover, and moisture availability. The densest populations are found on the Hornsby Plateau to the north and the Warrenora Plateau to the south, both of which are characterized by deeply dissected sandstone landscapes with shaded gullies, dense eucalyptus forests, and soils that retain moisture year-round. The dry, flat, sandy plains of Western Sydney and the Cumberland Plain, by contrast, support far fewer funnel webs. Their numbers increase again in the foothills of the Blue Mountains, where the terrain becomes rugged and moist once more. 3 funnel web species are common in the Greater Sydney region: the Sydney funnel web proper, the Southern Sydney funnel web, and the Southern tree-dwelling funnel web, Hadronich cerborea, which, as its name suggests, builds its retreats in the trunks and bark of trees rather than in the ground. Females are the master builders. A female Sydney funnel web may spend her entire adult life within or near a single burrow, maintaining it, expanding it, and defending it for up to 8 years or more. She rarely ventures more than a few body lengths from the entrance. Her world is the burrow and the small patch of ground that her trip lines cover. She is sedentary, patient, and extraordinarily long-lived for a spider. Males, by contrast, build burrows during their juvenile years, but abandon them entirely once they reach sexual maturity, around 4 years of age. After that, the male never builds another home. He wanders, and it is this wandering that brings him into contact with humans. The burrow itself is a masterpiece of microclimate engineering. Funnel webs require high humidity to survive. Their book lungs are efficient, but lose moisture rapidly in dry conditions. And exposure to direct sunlight and dry air can dehydrate and kill a funnel web in a matter of hours. The silk-lined burrow solves this problem elegantly. It traps moisture from the surrounding soil, creating a cool, humid microenvironment that remains stable even when the surface conditions above are hot and dry. This is why funnel webs favor south-facing and east-facing slopes, shaded gullies, and areas with dense ground cover. All locations where the soil retains moisture and the sunlight is filtered. It is also why funnel web populations are densest in the bushy, well-watered suburbs of Sydney's North Shore and the Warrenora Plateau to the south. Areas where sandstone ridges and moist gullies provide ideal burrowing habitat. In a curious correlation, the most expensive real estate in Sydney tends to overlap with the densest funnel web populations. The leafier and more heavily vegetated the suburb, the more funnel webs you are likely to find. Now, let us talk about the venom. The venom of the Sydney funnel web spider is one of the most studied and feared biological substances in the world, and for good reason. It is a complex cocktail of proteins, peptides, and small molecules. But the component that makes it lethal to humans is a single peptide, delta-atracotixin, also known as robustoxin. This is a 42 amino acid polypeptide, cross-linked by 4 disulfide bonds into a compact, remarkably stable three-dimensional structure known as an inhibitor cysteine knot, or ick motif. This molecular architecture is shared by a wide range of spider toxins and is one of the most ancient and successful protein scaffolds in the venomous world. Delta-atracotixin works by targeting voltage-gated sodium channels, the tiny protein gates embedded in the membranes of nerve cells that control the flow of sodium ions in and out of the cell. Under normal conditions, these channels open briefly when a nerve fires, allowing sodium to rush in and generate an electrical impulse. And then they rapidly close or inactivate, resetting the cell for the next signal. What delta-atracotixin does is prevent this inactivation. It binds to a specific region of the sodium channel, the so-called Site 3 receptor, on domain 4 of the channel's voltage sensor, and it locks the channel in its open position. The result is catastrophic. With the sodium channels stuck open, the nerve cells fire continuously, uncontrollably, over and over again, sending a cascade of electrical signals racing through the nervous system. This uncontrolled neural firing triggers a massive, simultaneous release of neurotransmitters from both the autonomic and somatic nerve endings. Acetylcholine, norepinephrine, and epinephrine flood the synapses. The body goes into what can only be described as a total systems overload. The muscles begin to twitch and fasciculate. Blood pressure spikes violently, then crashes. The salivary glands go into overdrive, The eyes stream with tears, the lungs flood with fluid. The heart races, then stutters, then begins to fail. In the most severe cases, the victim can progress from initial bite to complete respiratory and circulatory collapse in a terrifyingly short window. In small children, death has been recorded in as little as 15 minutes after the bite. In adults, the average time to death before antivenom was available was approximately 3 hours, though some victims survived for up to 3 days in intensive care before succumbing. Here is where the story takes one of its strangest turns. This venom, this extraordinarily efficient human killer, is almost entirely harmless to most other mammals. Dogs and cats show virtually no symptoms after a funnel web bite. Rabbits are unaffected. Rats require massive, experimentally administered doses before they show any toxic effects at all. The venom seems to have an almost laser-like specificity for primate nervous systems. Humans and monkeys are exquisitely sensitive to delta-atracotixin. Most other vertebrates are not. For decades, this bizarre selectivity baffled researchers. Why would a spider that evolved in Australia, a continent that has never had native primates, produce a venom that is specifically lethal to primates? The answer, published in a landmark 2020 study in the Proceedings of the National Academy of Sciences, is that the lethality to humans is an evolutionary accident. The researchers, led by a team from the University of Queensland and James Cook University, sequenced the venom gland transcriptomes of 10 different funnel web species and identified 22 distinct delta hexatoxins, the broader family of toxins to which delta atracotixin belongs. Their analysis revealed that these toxins are extraordinarily ancient, having remained remarkably conserved over the past 150 to 200 million years of funnel web evolution. Far longer than primates have existed, the researchers proposed that delta hexatoxins were originally developed as insecticidal compounds, used by juvenile spiders and females to subdue insect prey such as cockroaches, beetles, and flies. But as male funnel web spiders evolved their distinctive mating behavior, leaving the safety of their burrows to wander in search of females, they became far more vulnerable to vertebrate predators, birds, lizards, dunarts, bandicoots, and other small insectivores that would happily make a meal of a wandering spider. Natural selection favored males whose venom could deter these predators, not by killing them necessarily, but by causing intense pain. The researchers demonstrated that delta atracotixin activates specific pain-signaling sodium channel subtypes in mice, causing a pronounced pain response. A predator that bites down on a funnel web and is rewarded with a mouthful of searing agony is very likely to spit the spider out and never try again. The fact that this same venom happens to be catastrophically toxic to primates is simply an unhappy side effect of the molecular similarity between the sodium channels of funnel web predators and those of humans. As one of the researchers put it, the spider's ability to kill us predates our very existence. It is nothing personal. It is just spectacularly bad luck. And the difference between male and female venom drives this point home. Female Sydney funnel webs, which spend their entire lives in the relative safety of their burrows and rarely encounter vertebrate predators, produce significantly less delta-atracotixin than males. Their venom is estimated to be roughly 6 times less toxic to primates. No severe envenomation by a female Sydney funnelweb has ever been recorded. Every single fatal bite where the sex of the spider could be determined was inflicted by a male. The venom's lethality is not a weapon for hunting. It is a weapon for survival during the most dangerous period of a male spider's life, the mating season. During the warmer months, typically from late spring through early autumn, sexually mature male Sydney funnelwebs leave their burrows and begin wandering in search of receptive females. This is a perilous journey. Males are smaller than females, lighter, and more lightly built. They are exposed to dehydration, predation, and the very real risk of being killed by the female they are trying to mate with. Their wandering is primarily nocturnal because daytime surface conditions, the heat, the dry air, and the UV radiation would quickly dehydrate and kill them. At dawn, after a night of searching, they must find a cool, dark, moist place to hide until the next nightfall. This is where the trouble starts. The cool, dark, moist places that a wandering male funnelweb is looking for are exactly the kinds of places that humans create in their suburban environments. Garages, garden sheds, laundry rooms, piles of shoes left by the back door, swimming pools, especially swimming pools. Male funnelwebs are attracted to water, and they frequently fall into backyard pools during their nocturnal wandering. What makes this especially alarming is that they do not drown. Funnelweb spiders can survive immersion in water for up to 24 hours by trapping a layer of air in the fine hairs covering their abdomen. A spider pulled from a swimming pool is not a dead spider. It is a wet, angry, fully venomous spider. And more than one bite has been inflicted on a homeowner who reached into the pool to remove what they assumed was a drowned insect. The defensive behavior of a disturbed funnelweb is one of the most striking displays in the arachnid world. When threatened, the spider does not retreat. It does not flee. It does not play dead. Instead, it rears up onto its hind legs, lifting the front of its body high off the ground, and spreads its chelicerae wide, displaying both fangs in their full, glistening, downward curving glory. This is not a bluff. If the threat does not withdraw, the spider will lunge forward, driving those fangs into whatever is in front of it with startling speed and force. Funnelwebs have been described as some of the most aggressive spiders in the world. Though defensive might be a more accurate term, they are not seeking out confrontation. But when confrontation finds them, they meet it head-on with every weapon they have. It is worth addressing a common myth here. Funnelweb spiders cannot jump. This belief is surprisingly persistent in Australian folklore, and it is understandable. The speed with which a disturbed funnelweb can rear and strike can give the impression of a leaping attack, especially when the spider is elevated on a wall or fence, and gravity aids its forward motion. But there is no anatomical basis for a true jump. Funnelwebs can run quickly over short distances, and they can strike with considerable reach, but they are ground-bound. They also do not chase people. A funnelweb that has been disturbed will typically hold its defensive posture until the threat has passed and then seek cover. The spider is not hunting you. It is defending itself. The male's mating behavior itself is a high-stakes operation. When a wandering male detects the pheromones of a receptive female, he approaches her burrow cautiously, tapping on the silk entrance to announce his presence. If the female is receptive, she will allow him to approach. If she is not, she will attack, and the male must flee or be killed. During mating, the male uses the tibial spur on his second pair of legs to brace open the female's fangs, preventing her from biting him while he inserts his palpal organs, the modified leg tips that function as his reproductive organs, into her genital opening to transfer his sperm. The entire process is tense, rapid, and frequently fatal for the male, who may be killed and eaten by the female immediately after mating. Even if he survives, his remaining lifespan is short. Males typically die within a few months of reaching maturity, exhausted by the demands of wandering and mating. Females, by contrast, can live for 8 years or more when the fangs of a male Sydney funnel web connect with human flesh. The consequences can be dramatic. The bite itself is immediately and intensely painful, not because of the venom, which takes a few minutes to begin its work, but because of the physical trauma inflicted by the fangs themselves. Those chelicerae are large, sharp, and driven with considerable force. The fang marks are usually clearly visible, separated by several millimeters, and local bleeding is common. In some cases, the spider will remain attached, grip locked, jaws working, delivering multiple bites before it can be dislodged. If significant envenomation occurs, the symptoms develop rapidly. Studies have found a median onset time of just 28 minutes. The initial signs are local and systemic: tingling and numbness around the mouth, twitching of the tongue and facial muscles, profuse sweating, goosebumps across the skin, drooling, and watery eyes. The victim's heart rate increases. Blood pressure rises sharply. Then, the more alarming symptoms begin. Muscle fasciculations spread from the face to the intercostal muscles, the muscles between the ribs that control breathing. Nausea, vomiting, and abdominal pain set in. The pupils dilate. The victim becomes agitated, confused, and may become increasingly combative before sinking into unconsciousness. In the most severe cases, the final stages involve pulmonary edema, flooding of the lungs with fluid, profound hypotension, a collapse in blood pressure, and respiratory failure. Without medical intervention, death follows. Children are at particularly high risk. A study of funnel web envenomation cases found that 42% of all severe envenomations were in children. Their smaller body mass means a relatively larger dose of venom per kilogram, and their physiology is less able to compensate for the cardiovascular and respiratory insults caused by the toxin. One small child died within 15 minutes of being bitten. This is not a spider that offers second chances. And yet, since 1981, not a single person has died from a Sydney funnel web spider bite. Not one. This is one of the great success stories in the history of medicine. And it is a story that revolves around a man named Struan Sutherland, a herd of rabbits, and one of the most improbable antivenom programs ever conceived. Struan Keith Sutherland was born in Sydney in 1936 and grew up in Bendigo, Victoria. He studied medicine at the University of Melbourne, served in the Royal Australian Navy, and in 1966 joined the Commonwealth Serum Laboratories, or CSL. where he was appointed head of immunology research. It was a position he would hold for 28 years. And from that position, he would save more lives than most physicians could dream of. Sutherland's work on funnel web antivenom was driven by tragedy. In January 1979, a 31-year-old woman named Christine Sturges died from a Sydney funnel web spider bite. Her death galvanized Sutherland, who had already been developing the pressure immobilization first aid technique for snake bites, into action. Previous researchers had tried and failed to develop a funnel web antivenom. The conventional approach, injecting venom into horses and harvesting the resulting antibodies, did not work well with funnel web venom. Sutherland and his team took a different approach. They used rabbits. The process was painstaking and dangerous. First, venom had to be collected from live male funnel web spiders, a task that involved delicately stroking the fangs of one of the world's most aggressive spiders and collecting the tiny droplets of venom that appeared. This venom was then injected in tiny, carefully calibrated, gradually increasing doses into rabbits. Over a period of about 6 months, the rabbits' immune systems generated antibodies against the venom's toxic components. Blood was drawn from the rabbits, and the antibody-rich serum was purified into a usable antivenom. In January 1980, just a year after Sturges's death, another tragedy struck. 2-year-old James Cully was bitten by a Sydney funnel web and died 3 days later. He was the 13th confirmed victim. It was the last. Later that year, Sutherland's team produced a viable antivenom. Its first clinical use came on January 31, 1981, when a 49-year-old Sydney man named Gordon Wheatley was bitten by a male Sydney funnel web spider. He was treated with the new antivenom and made a complete recovery within 2 days. Since that day, no person has died from a confirmed Sydney funnel web spider bite. The antivenom has proven effective not only against the Sydney funnel web, but against all other Australian funnel web species, and even against the venom of the related eastern mouse spider. It is one of the most broadly effective antivenoms ever produced. The standard treatment protocol calls for an initial dose of 2 vials of antivenom, with 4 vials recommended if symptoms of severe envenomation are present. Patients are reassessed every 15 minutes, and additional vials are administered if symptoms do not resolve. In most cases, 2 to 4 vials are sufficient, but the record stands at 12. In February 2017, a 10-year-old boy on the central coast of New South Wales was bitten on the finger by a male Sydney funnel web that had been hiding inside a shoe. The boy developed severe envenomation symptoms and required a staggering 12 vials of antivenom to stabilize him. He survived. It remains the highest recorded number of antivenom vials used to treat a single funnel web bite. The transformation in outcomes has been extraordinary. Before the antivenom, a severe funnel web bite meant an average of 14 days in hospital if the patient survived at all. Afterward, the typical hospital stay dropped to 1 to 3 days. The antivenom works quickly, neutralizing the circulating toxin and reversing the cascade of symptoms that would otherwise progress to respiratory and cardiac failure. Sutherland also established the effectiveness of the pressure immobilization technique for funnel web bites. This first aid method, originally developed for snake bites, involves wrapping the bitten limb firmly with a compression bandage and immobilizing it with a splint. The technique slows the movement of venom through the lymphatic system, buying critical time for the victim to reach a hospital. There is even some evidence that prolonged immobilization may partially inactivate the venom itself. Between the antivenom and the pressure immobilization technique, what was once a potentially fatal encounter has been reduced to a manageable medical event, typically requiring 1 to 3 days of hospital care. But producing the antivenom requires a constant supply of raw venom, and producing raw venom requires a constant supply of spiders. This is where one of Australia's most extraordinary institutions enters the story: the Australian Reptile Park. Located on the central coast of New South Wales, about 50 miles north of Sydney, the Australian Reptile Park is the only facility in Australia that milks funnel web spiders for their venom. It is the sole supplier of raw funnel web venom for the entire country's antivenom production. The park houses thousands of spiders, from tiny spiderlings to fully mature adult males, each one individually housed and cared for by a team of dedicated keepers. The adult males are milked on a regular schedule, roughly every 2 weeks, a process that involves gently coaxing the spider to expose its fangs, and then using a fine glass pipette to collect the minute droplets of venom that bead at the tips. Each milking yields only a fraction of a milligram of venom. One single dose of antivenom requires approximately 70 individual milkings. Producing enough antivenom to meet Australia's medical needs, roughly 300 treatments per year, requires thousands of milkings from thousands of spiders. The park relies heavily on public donations to maintain its spider population. Each year, particularly during the warmer months, when wandering males are most active, members of the public are encouraged to safely capture funnel web spiders they find in their homes and gardens and deliver them to the park or one of its designated drop-off locations throughout Greater Sydney. Egg sacs are especially valuable. A single funnel web egg sac can contain between 50 and 150 spiderlings, which the park's keepers raise to maturity over a period of 3 to 4 years before they enter the milking program. In 2019, the park established a formal breeding program to supplement public donations and ensure a reliable supply of spiders year-round. Perhaps the most famous spider in the park's history is a male named Hercules, who was donated in early 2024, measuring 7.9 centimeters from foot to foot. Hercules was the largest male Sydney funnel web ever recorded at the park, larger than a baseball. His size made him exceptionally valuable for the venom program. A spider that big can produce a substantial quantity of venom with each milking. As spider keeper Emma Tenney put it at the time, receiving a male funnel web that size was like hitting the jackpot. In January 2025, the scientific understanding of the Sydney funnel web spider was fundamentally rewritten. An international team of researchers from the Leibniz Institute for the Analysis of Biodiversity Change in Germany, the Australian Museum in Sydney, and Flinders University in Adelaide published a landmark study in the journal BMC Ecology and Evolution that established what had long been suspected. The creature known as the Sydney funnel-web spider is not a single species. It is a species complex, a group of 3 distinct species that had been lumped together under one name for nearly 150 years. The study used a combination of detailed anatomical comparisons and molecular DNA analysis to examine funnel-web populations from across the Sydney region. The results were striking. The classic Sydney funnel-web, Atrax robustus, the spider originally described by Octavius Pickard Cambridge in 1877, was found to be concentrated in the Sydney metropolitan area, particularly the leafy northern suburbs, from the Central Coast south to the Georges River, and as far west as Bockham Hills. A second species, Atrax montanus, the southern Sydney funnel-web, was identified as occupying a range that overlaps with Robustus, but extends further south and west into the Blue Mountains and the Southern Highlands. This species had actually been described back in 1914, but was later dismissed and folded back into Robustus. The new study resurrected it. And then there was the third species, the one that had been hiding in plain sight for years, donated to the Australian Reptile Park by members of the public, who simply thought they had an unusually large Sydney funnel-web. The Newcastle funnel-web, Atrax christenseni, found only in the vicinity of the city of Newcastle, about 150 kilometers north of Sydney, is the largest of the 3 species. It had been informally dubbed Big Boy by spider enthusiasts and reptile park staff, who had noticed that the biggest males in the Venom program always seemed to come from the Newcastle area. The study confirmed their hunch. Big Boy was not just a large funnel web. It was a separate species entirely. The new species was named Atrax christenseni in honor of Kane Christensen, a spider enthusiast and former reptile park staff member who had spent years collecting specimens and documenting the differences he observed in the Newcastle spiders. His meticulous observations, particularly his noting of the unusually elongated male reproductive organs in the Newcastle specimens, provided the initial clues that something taxonomically significant was going on. The practical implications of this discovery are still being explored. The existing antivenom produced from the venom of Atrax robustus remains effective against all 3 species, and indeed against all other Australian funnel-web species. But researchers suggest that understanding the biological differences between the 3 species, particularly any differences in venom composition, could eventually allow antivenom production to be optimized and refined. If the 3 species produce subtly different venoms with different proportions of active compounds, tailored antivenoms could potentially be more effective and cause fewer side effects. Perhaps the most remarkable chapter in the story of the Sydney funnel-web is the one that is still being written. The transformation of one of nature's most feared substances into a potential life-saving medicine. In 2021, a team of researchers from the University of Queensland, led by Professor Glenn King and Associate Professor Nathan Palpant, announced a discovery that could change the landscape of cardiac medicine, working with the venom of the Caigari funnel-web spider, Hadronich infensa, a close relative of the Sydney species. The team identified a small protein molecule called Hi1A. This molecule, isolated from one of the deadliest venoms on Earth, showed a remarkable ability to protect heart cells from the damage caused by heart attacks. Here is how it works. When a heart attack cuts off blood flow to a section of the heart, the affected cells are starved of oxygen. This oxygen deprivation causes the cellular environment to become dangerously acidic, a condition called lactic acidosis. In response, the heart tissue activates acid-sensing ion channels, specifically the ASIC-1A channel, which sends a chemical death signal to the surrounding cells, triggering a cascade of programmed cell death. A healthy heart muscle is destroyed not just by the initial blockage, but by the wave of cell death that follows. No drug currently exists that can block this death signal. No drug on the market can prevent the downstream damage caused by a heart attack. Hi1A can. The molecule blocks the ASIC-1A channel, preventing the death signal from being transmitted. In laboratory tests on human heart cells exposed to heart attack conditions, Hi1A dramatically reduced cell death and improved the survival of heart tissue. The implications are staggering. If Hi1A could be administered to a heart attack victim in the field, perhaps by a paramedic in an ambulance, it could prevent the cascade of damage that turns a survivable cardiac event into a fatal one. It could buy time for patients in remote areas where hospitals may be hours away. And it could extend the viability of donor hearts for transplantation. Hearts that stop beating for more than 30 minutes before retrieval are currently considered unusable. But if Hi1A could keep the heart cells alive for even 10 or 15 additional minutes, it could dramatically expand the pool of donor organs available. The research has progressed rapidly. In 2022, a company called Infensa Bioscience, co-founded by King and Palpant, raised $23 million in seed funding to develop Hi1A into a clinical drug. Preclinical tests published in 2024 showed that Hi1A met critical safety and efficacy benchmarks, performing as well as the only cardioprotective drug to have ever reached advanced clinical trials. A drug that was ultimately shelved due to side effects. Crucially, Hi1A was found to interact only with cells in the injured zone of the heart during an attack, leaving healthy tissue unaffected, a feature that significantly reduces the risk of side effects. The team is also exploring whether Hi1A can be adapted to treat stroke, where the same acidosis-driven cell death mechanism damages brain tissue. The challenge there is getting the molecule across the blood-brain barrier, an obstacle the researchers are working to overcome by miniaturizing the Hi1A protein while preserving its therapeutic effects. If they succeed, a molecule from one of the deadliest venoms on Earth could become one of the most important drugs in modern medicine. And this is not the only potential medical application of funnel web venom. Other compounds in the venom are being investigated as bioinsecticides, targeted pesticides that could kill agricultural pests without harming beneficial insects or the broader environment. Professor King won the Australian Prime Minister's Prize for Innovation for developing the world's first insecticides derived from spider venom. The same chemical arsenal that evolution spent 200 million years refining into a predator deterrent and prey capture system is now being repurposed by human ingenuity into tools for healing and agriculture. So where does that leave the Sydney funnel web spider? This ancient, glossy, fiercely armed survivor of mass extinctions and continental drift. This accidental primate killer turned potential lifesaver. In truth, the Sydney funnelweb is not a species in immediate danger. It is not listed as threatened or endangered. Its habitat, the moist forests and suburban gardens of the greater Sydney region, remains abundant, and the spider's populations appear stable. Funnelwebs are adaptable creatures. They thrive in the disturbed, irrigated, well-mulched gardens of suburban Sydney almost as well as they do in undisturbed bushland. In some respects, urbanization has been kind to them. Garden rockeries, compost heaps, watered lawns, and the moist soil beneath house foundations all provide excellent burrowing habitat. But the spider does face challenges. Urban development continues to fragment and destroy native bushland across the Sydney Basin, reducing the extent of the spider's preferred habitat. Climate change poses a more insidious threat. Funnelwebs are highly sensitive to moisture levels, and prolonged droughts, which are becoming more frequent and severe across southeastern Australia, can dry out the soil and make large areas of habitat unsuitable. Rising temperatures may also push the spiders into increasingly narrow pockets of suitable microclimate, particularly in the drier western suburbs and inland areas, where conditions are already marginal. Changes in rainfall patterns, both drought and the increasingly intense downpours that follow, could also disrupt the spider's reproductive cycles and the stability of their burrow environments. There is also the matter of misconception. Despite decades of public education, many Australians still react to funnelweb spiders with indiscriminate fear and hostility. Spiders found in homes and gardens are often killed on sight, and many other harmless species, black house spiders, trapdoor spiders, and even certain beetles, are frequently misidentified and destroyed in the mistaken belief that they are funnelwebs. While killing individual spiders has little impact on overall population numbers, the broader attitude of reflexive extermination undermines the more nuanced understanding that these animals deserve. Funnelwebs are predators of garden pests, particularly cockroaches and beetles. They are prey for birds, lizards, centipedes, and parasitic wasps. They are part of an ecological community, and removing them does not make that community healthier. The silk of the Sydney funnelweb, incidentally, has its own history of human utility. The incredibly fine, uniform strands produced by these spiders were once used as crosshairs in optical instruments, including telescope eyepieces and rifle scopes. The silk's combination of extreme fineness, high tensile strength, and consistent diameter made it ideal for precision optics long before synthetic alternatives were available. The 2025 discovery that the Sydney funnelweb is actually 3 separate species adds another layer of concern. If the Newcastle funnelweb, Atrax christenseni, is restricted to a relatively small range around a single city, it may be more vulnerable to habitat loss and environmental change than previously thought. Each of the 3 species may have different conservation needs, and managing them effectively will require a much more detailed understanding of their individual ranges, population sizes, and ecological requirements than currently exists. And then there is the cultural dimension. The Sydney funnelweb has been a part of the city's identity for nearly a century. Ever since that first fatal bite in 1927, it is feared, yes, but it is also respected, studied, and in its own dark way, beloved. The Venom milking program at the Australian Reptile Park is a source of genuine national pride, a collaboration between scientists, zookeepers, and ordinary members of the public who catch spiders in their gardens and drive them to drop-off points, all in the service of saving human lives. It is a relationship built on knowledge rather than fear, on coexistence rather than extermination. The funnel web's ancestors walked this ground when the world was a different place entirely, when Australia was still locked into the belly of Gondwana, when the forests were full of creatures that no longer exist. This spider has survived mass extinctions that wiped out the dinosaurs. It has endured ice ages and continental collisions, and the slow, grinding reshaping of the landscape over geological time. And now, in the most improbable of developments, the venom that once killed 13 people is being transformed into a substance that could save millions. There is a kind of dark poetry in that, a spider that no one wanted to share a continent with, a spider whose very name makes people flinch, turns out to be carrying, in the tiny droplets that glisten at the tips of those terrible fangs, one of the most promising cardiac drugs the world has ever seen. Nature, it turns out, does not deal in simple categories of good and evil, dangerous and safe, useful and useless. The Sydney funnel web is all of those things at once, a relic of the deep past, a danger of the present, and possibly a key to the future. So the next time you hear about someone in Sydney pulling a big, black, glossy spider out of their swimming pool, spare a thought for the creature at the other end of the glass jar. It has been here for 200 million years. It was never meant to hurt us. And one day, it might just save your life. For the roughly 30 to 40 people who are bitten by Sydney funnel web spiders each year, the experience remains frightening. The pain is real. The symptoms are dramatic. The trip to the emergency room is urgent. But because of the work of Struan Sutherland and his team, because of the keepers at the Australian Reptile Park who milk spiders every day, because of the ordinary Sydneysiders who catch spiders in their gardens and drive them to drop-off points, the outcome is almost always the same: a few days in hospital, a good story to tell at dinner parties, and a healthy respect for the ancient creature that shares their city. The funnel web does not care about our fear. It does not register our fascination. It sits in its burrow, front legs resting on the silk, reading the vibrations of the world above, waiting for a beetle or a cockroach to stumble across its trip lines. It has been doing exactly this for longer than the Andes have existed, longer than the Himalayas, longer than grass. It is not impressive despite being a spider. It is impressive because it is a spider, because it has taken the most basic toolkit that evolution offers, silk, venom, and patience, and refined it into something that has outlasted nearly everything else on this planet. The Sydney funnel web is not a monster. It is a survivor. And in the strange, interconnected way that nature works, it is also, possibly, a healer. That glossy black body, those terrible fangs, that venom that can stop a human heart, they are all part of a story that began in the ancient seas of the Paleozoic and is still being written in the laboratories of Queensland and the emergency rooms of Sydney. It is a story about fear and fascination, about death and medicine, about the astonishing ways in which the most dangerous things in nature can turn out to be the most valuable. Sleep well.